Hi everyone. It is my pleasure to introduce to you our first speaker of the day, Dr. Lilit Nersisyan. Dr. Nersisyan earned her PhD from both the Institute of Molecular Biology in Armenia and Leipzig University in Germany in 2017. Her doctoral research revolved around developing computational approaches to investigate telomere length and sequence regulation, focusing on the roles these elements play in aging and cancer. In 2021, Dr. Nersisyan returned to Armenia, where she became the founding director of the Armenian Bioinformatics Institute, ABI. Under her leadership, ABI was established as a crucial scientific educational foundation created to address the shortage of bioinformatics expertise in Armenia. She now leads research teams at both the Armenian Bioinformatics Institute and the Institute of Molecular Biology, continuing to drive groundbreaking research at the intersection of computational biology and human health. Please join me in welcoming Dr. Lilit Nersisyan.
Thanks a lot, and thank you all for being here. I'm very excited to share the story of the Armenian Bioinformatics Institute with you. The first time I had to share this story with the international community was on a TED platform, and then I was hesitant because I was thinking that this is a story about Armenia and would only be interesting to Armenians. However, it appeared that it actually resonated with many people who were looking to bring their knowledge and expertise back to their home countries. And in a sense, this is also a story about an attempt for science democratization and for making bioinformatics more accessible to more people. And I think life sciences needs more bioinformatics. One of the arguments for this is the amount of data. You can see on the upper curve the amount of data that is annually deposited in the European Genome Archive. And what you can appreciate is that this y-axis is logarithmic, which means that data is growing exponentially. Not only is the amount of data a challenge for bioinformatics, but also the complexity. So here you can see the number of articles in PubMed that use different data modalities from genomics to transcriptomics to microbiome. And you can appreciate that the complexity of the data increases as well. So bioinformatics needs to analyze all this data in an integrative manner to infer knowledge. And what you can also see is that the growth here is linear, it's not exponential, which gives me some grounds to speculate that perhaps bioinformaticians could do a bit better task at extracting knowledge from the existing data. And there's a lot of work for us bioinformaticians to do, and a paper in 2019 has shown that there was a 50% negative employment rate in bioinformatics, both in academia and industry, meaning that half of the bioinformatics positions are not being filled. And I imagine that this number might have grown during the last years. And the situation is, you can imagine, not better in Armenia, but before jumping to this story, I would like to introduce the country shortly. It's a very small, cute country, you can see, located in the Caucasus region between Georgia and Iran. It has only 3 million inhabitants. This is the capital city, Yerevan, where I grew up. Armenia is surrounded and has a lot of mountains, and what it is best known for among the tourists is the vast amount of old churches as one of the oldest Christian countries. Armenia has been part of the Soviet Union, and in the 90s, when the Union collapsed, after the 30 years that followed, unfortunately science wasn't prioritized by the government as much. In 2021, the science funding was at 0.25% of GDP. In comparison, in most of the Western countries, it is more than 1 or 2% that is devoted to science. And correspondingly, the number of researchers has declined drastically from 50,000 during the Soviet Union to less than 5,000 survivors until now, and half are at the retirement age. In the recent years, the government has started increasing the amount of funding, so there is optimism and a shift of directionality in this direction. And several specific research fields still managed to survive and even thrive, but this was more of a sporadic phenomenon and a result of the effort of individual researchers. And the same was about bioinformatics. So in 2000, several research groups were established that were doing work on structural bioinformatics. And it was not until 2011 that the first group was established that was focusing on the analysis of genomics data. It was established by Arsen in the Institute of Molecular Biology of the National Academy. I was his first student, and for many years it was only me and him in the team, and then other people started joining in. And an important collaboration we established was with Hans, who was the managing director of the Bioinformatics Center at the University of Leipzig at the time, and this collaboration helped us a lot to establish the field in Armenia. So we were doing research in systems biology and in telomere biology that my thesis was focused on. I then went to Sweden, to Karolinska Institute, and got a Marie Curie Fellowship to study mRNA degradation. And in 2021, I was thinking of going back to Armenia and establish my own team in IMB. However, I faced a nice challenge in that there was nobody to hire, basically. And if you see, after the establishment of the group in 2011, 10 years afterwards, there were only 2 PhD students in Arsen's team, and no other research group had emerged that would focus on genomic data analysis. And the reason for this is basically the broken ecosystem. Educational programs not providing specialists at an adequate level, the research infrastructure not there. Arsen, Hans, and all of the team were working hardly to convince the government to support the establishment of a sequencing facility, and at the time it was thought to be a very complex job with no specialists in place. And basically with no infrastructure and a virtually non-existent modern biotech industry, this ecosystem was not functioning well. So instead of just establishing another research group, we had to fix this. Talking to the state, it was clear that bioinformatics is not the #1 priority because there were lots of other things for the state to solve at the moment. And following the example of the European and other countries, we decided to arrive at a crazy idea of establishing a bioinformatics institute ourselves. So in 2021, we launched the institute as a scientific educational foundation, and our mission was to train leaders in genomics and bioinformatics who would be in capacity to then go and modernize research, education, biotech, and biomedical industries. At first, this seemed quite a challenging task and a bit confusing because it was not apparent who to talk to, where to start from. Our starting capital was $10, a symbolic amount of money. And in Armenia, there were 2 people with a PhD degree in bioinformatics, including myself and Arsen. So this task was resembling more the challenge that Noah had in front of him when he tried to reproduce a set of species with only 2 individuals in each. And I guess bioinformatics was even as challenging. And as scientists, when we have a hard challenge in front of us, we tend to divide and conquer. So retrospectively, I have divided the whole endeavor into 7 easy steps of how to establish a bioinformatics institute. So you can try this at home if you want to. It's very easy. So what we do, you start with a core team, and then you engage the international community. Afterwards, you have to find seed funding. And when I'm telling about seed funding, is what I learned the hard way is that when you ask people to give you money for establishing an institute, they think you're crazy. So instead of telling them that you want to establish an institute, you just ask for money for training or to support some students with scholarships. So you start small, and this is what we did. And then after we had enough capacity to show for, then we already went for more substantial funding to support our operations. The first thing we did is to organize a very intensive course in bioinformatics. And the course was meant for anyone with any background, basically, to give them the minimum amount of skills so that they are able to perform bioinformatics tasks, and so that by the end of the course, they are able to start their first bioinformatics project. After this course was over, we hired almost half of the cohort to ABI, and we engaged them into real-world research projects. And these projects were also partly supervised by our international community to make our job a bit easier. And then afterward, we already grew in capacity and had the critical mass of people so that we were able to form formal research units. So no remote supervision was needed afterwards. And what's important is that you repeat these steps from 5 to 7 every year to ensure continuity and sustainability of this effort. So I will guide you through each of these steps. The core team was created. First of all, it was the board of trustees. Hans was the chairman, and Arsen and Aram from Flagship Pioneering also joined the team. And an important contribution was made by the 2 PhD students of Arsen in organization of these courses and mentorship of the students afterwards. We had a team of advisors from different sectors, not only research, but also biotech, marketing, finances, everything. I'd like to mention Jonathan from the University of Pennsylvania. He, together with the board and the international scientific community, was joining remotely our group meetings and discussions. And it was important because in the last 5 or 10 minutes of the presentations, they would ask questions and give feedback, and basically show the students what science is all about. And for me, it is about discussions, brainstorming, and that's where innovation takes place. A lot of volunteers were helping ABI in the beginning, and we had around 50 people, either Armenian scientists or non-Armenian scientists from different parts of the world, helping us with the training and with the mentorship. And what I understood is basically, if people believe in your cause, they will devote the most expensive thing that they have, is their time. And this is what this community did for us. However, there is still a lot of effort that you have to put in order to consolidate these resources and manage their time wisely, and also to keep the momentum going after the initial wave of excitement is over. And time is very expensive, but we still needed proper money. So in terms of funding, it was again a bit challenging. Initially, we were relying on donations, either from organizations or from individuals. And then the funding sources diversified. So we could manage some grants from private foundations in Armenia and also some governmental grants as well. A bit challenging for us is that ABI is a private foundation, so it is not eligible to the majority of the governmental grants. And we also try to apply to the European grants, but they're even more challenging. And we have, for example, tried the Horizon Twinning grants 3 times in a row. We go past the threshold, however, still not lucky there, but we will continue pushing for this as well. And as you can see, the growing amount of funding sources comes actually from biotech companies located in the United States, UK, and Europe. And we provide bioinformatics data analysis services to them. It is relatively easy to find such collaborations because such companies outsource their bioinformatics a lot, and everyone needs bioinformatics. However, such collaborations have both good and bad sides. So the good opportunity is that this is one of the ways to ensure a remote semi-presence of these biotech companies in Armenia. And the hard thing is, our mission is to train independent scientists and train leaders. And the most of the staff at ABI are actually undergraduate students. But we have to put them on the independent research track early on. So they independently pursue their research stories as much as possible. They start writing grants early on, even from bachelor years, they start writing their manuscripts. And this requires a bit of patience from the management perspective. And industry doesn't like patience. Industry likes deliverables and strict timelines. And this is something that can disrupt the group dynamic a bit. So we have to be wise about choosing who we collaborate with and how we distribute our resources so that science and also our mission doesn't suffer on the way. But still, this is a very nice support to our initiative. An important activity for ABI is the organization of different training courses and summer schools. So we have different specialized courses in molecular biology, bioinformatics, But these summer schools are very important, and we named them omics schools in genome bioinformatics. The first one was organized in 21, and then it became an almost annual event. They last for 10 weeks, 6 hours a day, so it is an almost full-time engagement. And people who admit to these schools are usually bachelor students, sometimes higher or researchers from biology or from data and computer science major. And what we teach them is we start with molecular biology, statistics, programming. By this time, they all start speaking the same language. And then we go on to NGS technologies, algorithms, and different topics in functional genomics. And aside from delivering this systematic knowledge, we also invite speakers from different parts of the world to share their research projects and to kind of showcase to the students what the career in bioinformatics can potentially hold. And the school ends with their first bioinformatics projects. They spend several days outside of Yerevan working on their projects, making discussions, and then making their final presentations, and this is when the school is over. An important impact of the school is that after the school, the majority of people continue their careers in bioinformatics. Before coming to the school, they have most of the times no idea what bioinformatics is about. Some are employed by ABI, some go to different research institutes in Armenia, some to biotech industries. And what's also nice is that in the school, the ABI research staff, which is mostly formed by the graduates of the previous schools, is taking over the responsibility of mentoring the new students. And this also gives them the opportunity to have this teaching and mentorship skills developed, and also puts ABI on a relatively sustainable path. So currently we have formal research units. One is remotely supervised by Hans from Germany. The other one is supervised by me, and we also have a unit for collaboration with a biotech company. There are several challenges. I will mention only two. The first is the lack of senior scientists. So at the moment we have 4 people at their PhD level, and we have a difficulty hiring postdoctoral scientists from abroad yet, considering that we are a new institute. And this is a bit different how you manage projects. Usually as a principal investigator, you put the project on a postdoc or a PhD. In this case, there is a chance that the student gets admitted to a top university abroad for their masters, and they have to leave the project. And so what you have to do is, after the first year of training, the student starts contributing to the project, and then you have to make sure that the project is handed over to the next student so that it doesn't suffer when people leave. And what I call this process is managing projects with a temporally distributed grid of undergraduate students. It's a challenge, but it's possible. It makes projects a bit hard, but I guess we are dealing with it. And there are also a lot of opportunities, and one of it is that it is relatively easy to hire. So if you're a new PI or an established PI looking to expand bioinformatics expertise, you don't have to find funding for 3 to 5 years to support a PhD student. You can start small with an intern and then grow from there. And why this is possible is because there is a flexibility of the funding schemes, so we are not very tied by all the bureaucratic procedures. And something I'd like to mention here as well is that Armenia is actually a nice source of biodiversity, so it's possible to get some unique data sets for interesting projects. So this is what we have achieved in numbers in the last 3.5 years. We have currently 3 research units with 6 grants and 3 industry collaborations. We have trained over 100 students, and we have hired over 30. And you can see that actually the research staff is converging at this number, because this is as many undergraduate students that 2-3 principal investigators can handle. So hopefully hiring another PI will be another way for us to grow in the future. And the number of publications is growing linearly at the moment. An important thing, we have secured funding to establish a powerful compute node for ABI. And finally, with the great efforts from Arsen at IMB, finally he convinced the government to provide some funding to establish the first NGS facility. And the first sequencing run was done the last month. And I would like to spend just a couple of minutes to introduce the research focus that ABI's labs have. So Hans's team does cancer genomics. And they try to find, they explore cancer heterogeneity and try to interpret the genetic basis of heterogeneity of disease development and treatment response. They usually start with a machine learning algorithm called self-organizing maps on genomics, transcriptomics, or epigenomics datasets. And this algorithm produces such nice maps for each sample. And these are maps of co-regulated gene clusters colored for each sample individually. And they use these maps to cluster the samples and then identify different cancer subtypes and also possible transitions between those subtypes. Currently, they work on colorectal cancer subtypes and they map those to the liver metastasis subtypes and then try to see how this subtyping is responsible for differential treatment response in these patients. Another project that they embarked on is plant genomics, particularly grapevine. So Armenia is actually a nice source of grapevine varieties that live in the different elevations. And there was a paper in Science where IMB was also participating that showed that there is a dual origin of different domesticated grapevines, one as a fruit, table fruit, and the other one for wine production. And the one for wine production was actually originating in the region in Caucasus, where Armenia and Georgia are currently located. And what Hans's team is doing is basically describing the genetic landscape of the evolution of grapevine and its cultivation. And we also have different grapevine varieties that are resistant to climate change. And they look into the genetic basis that make this resistance possible. And recently, for example, they have found a factor that's important for cold acclimation of a cultivated vine, Vitis vinifera. Our team, we do microbiome research, and it is a continuation of the postdoc project that I did in Karolinska Institutet in Belagiano lab. There we develop a technology that looks into mRNA degradation intermediates. We sequence these intermediates, and what this technology allows us to do is to understand bacterial response to stress, particularly to antibiotic treatment. And what is nice about this technology is that you can take a very complex microbiome community, apply a drug to it, and after 5 or 10 minutes treatment without the need for cultivation or isolation of specific species, you can tell which bacteria are happy and which bacteria are sad. So basically, you kind of identify direct responses in a complex microbiome community. And what our lab is now interested in is how these direct responses are then propagated through the interaction between species in the complex microbiome community, and eventually how this propagation leads to the shift in the microbiome composition over time, eventually leading to disease or some irreversible changes. This is a multi-year challenge, and we divide it into different subtasks. So currently we, for example, look into the response of honeybee microbiomes to pesticide treatment. And we also look into clinical isolates in different bacteria in Armenia. And another project is a continuation of my PhD in telomere biology. So we developed software to measure the length of telomeres and also to assess the activity of different telomere maintenance pathways. And what we currently are looking at is the presence of different telomeric patterns in the cell-free DNA fraction of the blood plasma. So this cell-free DNA fraction appears there either from healthy cells or when there is a tumor, then the tumor cells shed their DNA to the bloodstream. And people use this cell-free DNA as a biomarker for early detection of cancer or for monitoring of cancer development. Looking into the telomeric patterns is a bit of a challenge, but we have recently discovered some biomarkers that could be cancer specific, and hopefully we will be able to publish that in the near future. So I would like to thank all the organizations and people who supported ABI in its establishment, and also to the students of IMPRS and all the organizers for organizing this conference and for inviting me here, and thank you for your attention.